Welcome, medical students, to today's histology lecture on the topic of renal histology. In this comprehensive script, we will delve into the intricate details of the renal nephrons, focusing on both cortical and juxtamedullary nephrons. As we progress through the script, we will explore the structure, function, histology, and regulatory mechanisms of these nephrons. By the end of this lecture, you will have a deep understanding of renal histology and its crucial role in maintaining overall health. Let us begin our journey of discovery. To understand renal histology, we must first grasp the concept of cortical and juxtamedullary nephrons. Cortical nephrons are primarily located in the outer cortex of the kidney, whereas juxtamedullary nephrons extend deep into the medulla. These two types of nephrons differ in their respective roles and anatomical features. While cortical nephrons make up the majority, around 85%, of nephrons, juxtamedullary nephrons play a crucial role in establishing and maintaining the renal concentration gradient. Now, let us explore the specific structure and function of the proximal convoluted tubule, PCT, in cortical nephrons. The PCT reabsorbs water, glucose, amino acids, and electrolytes from the tubular fluid. It is lined with cuboidal epithelial cells that have microvilli. The PCT also has mitochondria for energy. Next, we will discuss the histology of the loop of Henle in cortical nephrons. The loop of Henle, found in cortical nephrons, is important for urine concentration. It has two parts, the thin descending limb and the thick ascending limb. The thin limb has flat cells with few mitochondria. The thick limb has cuboidal/columnar cells that remove sodium, potassium, and chloride ions from the tubular fluid. This helps establish the renal concentration gradient. We will cover the DCT in cortical nephrons next. The distal convoluted tubule, DCT, is responsible for fine-tuning the reabsorption and secretion processes initiated in earlier segments of the nephron. This segment plays a crucial role in electrolyte handling, particularly sodium and calcium reabsorption, while actively secreting hydrogen and potassium ions. The DCT's epithelial cells are cuboidal in shape and possess fewer microvilli than the PCT. As we progress further, we will now explore the intricate blood supply and microcirculation within cortical nephrons. In this section, we will explore the complex blood supply and microcirculation in cortical nephrons. This includes the role of the afferent and efferent arterioles, the specialized glomerular capillaries, and the initial filtration process in the Bowman's capsule. Juxtamedullary nephrons are found in the deep cortical region, adjacent to the medulla. These nephrons are characterized by their unique anatomical adaptations that enable a greater concentration gradient establishment. They play a crucial role in regulating water balance and blood pressure. The juxtamedullary nephrons possess long loops of Henle, extending deep into the medulla, which aids in establishing and maintaining the osmotic gradient within the interstitium. Now, let us delve into the role of the juxtaglomerular apparatus in juxtamedullary nephrons. The juxtaglomerular apparatus, JGA, is crucial for regulating blood pressure and renal blood flow. It comprises macula densa, granular cells, and extraglomerular mesangial cells. Macula densa senses changes in fluid composition while granular cells secrete renin to counter low blood pressure. Extraglomerular mesangial cells provide structural support and communication. Let's now focus on how the renal concentration gradient is maintained in juxtamedullary nephrons. In the video, we will discuss the role of juxtamedullary nephrons in establishing and maintaining the renal concentration gradient. We will explore the countercurrent multiplier system and the interaction between the descending and ascending limbs of the loop of Henle. We will also highlight the significance of the vasa recta in water and solute exchange within the medulla. The vasa recta is a vital component of the juxtamedullary nephrons, serving as the capillary network responsible for maintaining the medullary concentration gradient. Structurally, the vasa recta has long, hairpin-shaped vessels lined by endothelial cells, which play a role in countercurrent exchange. In countercurrent exchange, the vasa recta allows for the transfer of water and solutes out of the medulla and back into the bloodstream, while preventing the dissipation of the concentration gradient. This mechanism helps to conserve water and maintain urine concentration. Now, let us delve into the intricacies of the countercurrent multiplier system. In the video, we will discuss the countercurrent multiplier system in juxtamedullary nephrons. This system establishes a concentration gradient within the kidneys and involves the interaction between the descending and ascending limbs of the loop of Henle. Water is reabsorbed in the descending limb, while the ascending limb actively transports solutes. This mechanism allows for the production of concentrated urine. We will also compare the histology of cortical and juxtamedullary nephrons. Cortical and juxtamedullary nephrons have distinct histological characteristics that correspond to their roles in renal function. Cortical nephrons have glomeruli predominantly in the outer cortex with short loops of Henle, while juxtamedullary nephrons have glomeruli deeper in the cortex and long loops of Henle extending into the medulla. These long loops facilitate greater water reabsorption and enable the production of concentrated urine. Understanding the regulatory mechanisms of blood pressure and water balance by juxtamedullary nephrons is essential. Juxtamedullary nephrons play a vital role in the regulation of blood pressure and water balance. Through their intricate countercurrent multiplier system and the ability to produce concentrated urine, these nephrons contribute significantly to the conservation of water and maintenance of electrolyte balance. Additionally, by modulating the secretion of renin, juxtamedullary nephrons participate in the renin-angiotensin-aldosterone system, ultimately regulating blood volume and blood pressure. The coordination of these processes ensures overall homeostasis within the body. Now, let us move towards the conclusion of our extensive lecture. In summary, the histology of renal nephrons, specifically cortical and juxtamedullary types, is crucial for maintaining overall health. These nephrons have unique adaptations and functions that help regulate electrolyte balance, blood pressure, and urine concentration. Understanding renal histology is vital for diagnosing and treating renal diseases.